She was a budding scientist in 2013, and for the good of Philippine science, she stayed for good. Fascinated by neuronal development and early animal evolution, she's also interested in regulatory networks that control gene expression dynamics, underlying the ability of many marine organisms to rapidly respond and adapt to their environment. So she'll speak on sponge microbiome dynamics under stress. Dr. Conaco.
This morning I will be telling you a little about one particular project that we've been doing in the lab as sort of an example of the types of questions that you could address using omics approaches if you were interested in microbes and the organisms that they interact with. So essentially, I'm going to give you a little crash course on sponge and microbiome diversity, the impacts of stress, as have been discussed earlier. And then I'll show you some of the data that we've amassed using some omics approaches to look at the sponge and its microbiome interaction. And then a little bit of looking forward. So, coming from the Marine Science Institute, we always show something like this. This is our token slide to always try to remind everyone how important it is, our location, to the types of questions that perhaps we should be considering. So we're located within the coral triangle, shown here by the blue shading. And this is an area of mega diversity. So a lot of corals, a lot of fish, also a lot of other organisms like sponges that I will talk to you about today, and the microbes that are found within all of these organisms. So sponges, which are some of my favorite organisms, are animals, and they are some of the most early diverging sister groups of the other animals. So about 600 million years ago, the sponge group exploded, and there's such a high diversity of sponges all throughout. They are a very diverse group owing to all of these millions and millions of years of evolution, and there's really four major groups. The most common one, of course, are the demosponges. So these are the ones that you will see anywhere you go snorkeling. They're very common, they're very diverse, they all look very different. There's the hexactinelids, the deep water, cold water sponges, like the Venus flower basket, sorry. And then there's the homoscleromorph sponges, and there are the calcareous sponges. So these are very special sponges because they make calcium carbonate skeletal elements, just like, similar to what your corals might make, whereas all the other groups produce silica skeletal elements, or siliceous skeletal elements. So sponges are very underappreciated, but they are quite important in a lot of ecological processes. So they play a role in the cycling and recycling of nutrients. They provide food, like for your turtles. They provide habitat for fish and other organisms. They're also really important in consolidating your reef systems and fire eroding, so it can reshape an entire reef through the action of your sponges. And most importantly for this forum is that they are reservoirs of microbial diversity. So they have a lot of perhaps even novel microbes that have never before been studied. And these microbes really enrich or expand the ability of the sponges to work within their environment. So sponges live with a lot of microbes, as I said, and these microbes complement the metabolic abilities of the host. They can also provide additional metabolic capabilities that allow the organism to survive in perhaps not so ideal situations, like let's say eutrophic conditions or polluted areas. They provide protection and defense. They maintain the balance within the sponge host, and they provide the extended functional genetic repertoire that adds to the abilities of that whole organism. So the sum of a sponge, is really much more than its parts. Okay, so sponges are predicted to be resilient to changing ocean conditions. So in this paper published in 2018, they say sponges will be the kings in the oceans under climate change scenarios. And in fact, it's said that when coral reefs bleach and corals die, everything will turn into a sponge reef, although of course there's also algal-dominated reefs and so on, but the sponge biologists love their sponges. So all it's to say is that sponges are very resilient, they're adaptable, and they're so diverse that they can really, a lot of them can adapt to different conditions. So the ability of the sponge to survive under stress and not so ideal conditions is mainly because of this stability of the partnership with the sponge animal and its microbes. So there are different trajectories that can happen when the organism is stressed, just like you and your gut microbiome. Under stress, you can be resistant to whatever the perturbation is, and you retain stability. Or you could have a certain small change happening, but you can acclimate and adapt. So you have adaptation. On the negative side, if the change in your holobiont, your change in your relationship becomes unbalanced, you can have something that's called dysbiosis, and this can lead to disease. So essentially, it's an imbalance within the host and within the microbiome. So the question that we wanted to ask is, how would different sponge types respond to future ocean conditions? So as I mentioned, sponges are not so well studied, so we wanted to sort of look at two different groups of sponges. One is an example of a siliceous demo sponge. So this yellow guy here is Neopetrosia compacta. And down here is an example of a calcium carbonate calcareous sponge called Lucetta chagosensis. So they're both sort of yellowish. The characteristics of these two sponges is that one has a lot of microbes, it's high microbial abundance. The other is low microbial abundance. So we wanted to look at how the microbial community of these sponges would change in response to the stress, and how the host transcriptome profile would sort of also correlate with these changes in the microbiome. So Dr. Reyes already gave you a really nice overview of these methods. So these are what we use. 16S ribosomal RNA sequencing for microbial community profiles, and then we did RNA sequencing to look at the host transcriptome. Okay, so first off, let's look at the microbial community structure of these two sponges. So here I'm showing you relative abundance plots of the microbes within each of these sponges. So you don't need to know what any of these things here are, but just you can clearly see, right, there's more colors for the siliceous sponge compared to the calcareous sponge, and that means that the calcareous sponge has a lot more different types of microbes. Each of the colors represents a different group. So from this particular figure, we can already tell that the siliceous sponge is taxonomically more diverse and potentially more functionally diverse as well, as compared to the other sponge. We then stressed these sponges out. We put them into these different conditions shown here. Present-day conditions as a control, and then we had acidification, we had warming treatments, and then we had combination treatments based on the predictions of the IPCC. So these were simple experiments just exposing sponges to different conditions. So this is what happened to the siliceous sponge. Over the different treatments, you see it looks pretty happy. Oh, we tinuhog namin siya. But anyway, that's how we set them up in the experiment. So the siliceous sponge was happy, but the calcareous sponge in some of the treatments started to show some necrosis of its tissues. So right away we can see that at the very initial sort of assessment, the calcareous sponge is more susceptible to the stress. So the calcareous sponge, in fact, exhibited 75% mortality at the highest, most stressful condition, whereas the siliceous sponge only showed something like 3% mortality. It was so robust. When we looked at the microbiomes of these sponges under the treatments, we found, as expected, that the siliceous sponge did not change much. So this is the microbiome structure for the siliceous sponge. Present day, acidified warming, and then the RCP conditions. You'll notice that the patterns are almost similar across. Very few bacterial taxa actually change in abundance. Not much happened here. But when we look at the calcareous sponge community, we found slight changes. Like in the warming, you had an increase in Vibrios, also in RCP6, and then at RCP8, where you had the most extreme stress, you had a lot more other types of microbes suddenly became detectable within the microbiome. We looked at some of the species or the taxa that were present, and some of these were opportunistic and potentially pathogenic. So right away you can see that under stress, a holobiont, the sponge holobiont, could potentially have an imbalance in its microbiome, and this could contribute to what we saw as mortality in the sponge. So the next thing we wanted to see was whether the sponge is also trying to compensate or what for this change in its microbiome, or whether something was happening within the host sponge that resulted in the change in the microbiome. And so we applied the RNA sequencing method here with transcriptomics. And this graph simply shows you the number of differentially expressed genes in the silicious sponge on the left side and the calcareous sponge on the right side. So you'll notice there's so much more differentially expressed genes in the calcareous sponge. It seemed more reactive to the stress. The silicious sponge, of course, also had a lot more differentially expressed at the most stressful conditions. So they were reacting, but in slightly different ways. We then tried to look at the functions that were represented in these different genes that were changing in abundance in the sponges. So since we didn't have too many genes in the silicious sponge that changed, we could not really analyze, or I'm not going to show you the analysis for functions, but for the calcareous sponge, this is what we found. So this heat map here essentially shows you the functions which are listed here and how abundant or enriched they are in each of these different treatments, which are color-coded here. So for example, protein folding, which is involved in helping your proteins renature if they are denatured under high heat, is actually quite enriched in the warming treatment and in the other treatments with elevated temperature, which is as expected. We also saw other stress response pathways that were being enriched in some of the treatments. We saw immune-related pathways, and then tissue maintenance pathways like your collagen metabolic process, cell-cell adhesion, cell communication, were also becoming enriched under certain treatments. What we noticed though is that the most robust or the most extreme stress condition also resulted in sort of the most enrichment for certain types of cellular functions, whereas acidification didn't seem to. Well, there was some change in transcription, how it seemed to be more less stressful, I would say, to the sponge. So overall, we can see that different functions are affected by exposure to different conditions, and we can see this at the level of the transcriptome. We then tried to look at the correlation between the bacteria changes and the changes in the transcriptome for the two different sponge species. And what we found is that for the silicious sponge, the correlation was not so good, but for the calcareous sponge, as I think you've already seen from the previous slides, the correlation between the number of differentially expressed genes and the number of the differentially abundant OTUs or taxa was pretty high. So we were thinking, what would have possibly caused this, apart from all of the other changes that we saw in the sponge host. So it's already known that the sponges actually have a way of sensing bacteria in their environment. They have receptors, just like we do. And they use these receptors to sort of detect whether they want this bacteria as a symbiont, or whether this is a food bacteria, or whether this is a foreign bacteria and we should get rid of it. So we looked at some surface receptors. There's a whole bunch of them, but I'm only showing you a few examples. So the extracellular SRCRs, or scavenger receptors, are known to be important for sensing bacteria within the environment and for discriminating between food and symbiotic bacteria. And then the G protein coupled receptors, which are also really common receptors for sensing external, the external environment. So for the siliceous sponge, what these graphs are showing you anyway is the relative expression patterns of the different receptors in the present day treatment all the way up to the most stressful condition. So what you'll notice is for the siliceous sponge, the SRCR genes tended to stay sort of similarly expressed all throughout the different treatments. The patterns remain similar. Same thing with the G protein coupled receptors. They also stay more or less similar. But when you look in the calcareous sponge, for the SRCRs, there were some genes that actually changed in expression pattern. Some were more low expressed, some were more high expressed under the warming condition, the yellow hills here, and in the most extreme stress condition, the red hills up here. The G protein coupled receptors also showed a slight change, but not as obvious as the one we saw for the SRCRs. So this points out a potential way in which that might explain how the sponge microbial community actually suddenly changed under the stressors. It's because perhaps something within the host also changed, which disabled its ability to detect foreign versus food versus symbiotic bacteria. So some type of control mechanism that was lost. Obviously, these are all hypotheses that need to be further tested and to be further looked into. But these types of methods actually just sort of help you point the way to other areas that you might later on explore further. So this is the summary that we now have, our working summary. So these two different sponges under stress conditions react differently. The siliceous sponge maintains a very stable transcriptome profile. It expresses heat shock proteins for protection. It expresses antioxidants. And as such, it's able to maintain stability of its microbiome. Although we saw a few changes, eventually I think it was still able to acclimatize to the stress conditions and was able to survive the treatment. On the other hand, our calcareous sponge up here had differential regulation of stress and immune response functions, as we saw from the transcriptome. There was also some changes in its surface receptors. And we saw the effect of this on the microbiome, so there was some recruitment of opportunistic and potentially pathogenic bacterial taxa. This would lead to dysbiosis and imbalance within the community, and as we saw with our own two eyes, some tissue necrosis and mortality occurred. So what we've seen here is that sponges and their microbiome sort of represent different strategies for succeeding in marine habitat. So the siliceous sponge had a different composition, a different genetic capability that allowed it to survive the stress, while the calcareous sponge had a different microbial composition and different genetic complement that made it a little less able to withstand the different stressors that we applied. But what's important to note is that both the host and its microbial symbionts must contribute to the functional response to a changing environment, which makes it really important to look not just at actions. So all of these different sponges have evolved many millions of years, and they're so well suited to whatever habitats they encounter that I think this is something that people should, or something that people can actually look at. Not just in sponges, of course, but also in other organisms. They're really interesting relationships that are so underexplored at this point. Okay. And so finally, I would like to thank this project. It was a small study within the Picard-funded coastal acidification program. All the work was done at the Bolinao Marine Laboratory. And most of the work that I showed you today is the work of my master's student, Nino Posadas, with the help of Mike and Jake. So very young students can do this type of work. I'm sure we all can, with a little bit of sort of trying to learn the methods and learning from others. So it's really I think it's one of those areas where interdisciplinary collaboration helps, because people who know the ecology of the organism, the biology of the organism, really enrich the types of questions and the types of approaches that we as microbiologists and molecular biologists, which I am, can use. So thank you very much for your attention.